Children, we had complete the lesson Biotechnology Principles and Processes. So today we can discuss some important questions from the lesson. Okay, so see the first question. How is the copy number of plasmid vector and yield of the recombinant protein related to each other? So I will read the question again. How is the copy number of plasmid vector and yield of protein related to each other? Answer. The copy number of plasmid vector is directly related to yield of recombinant protein, isn't it? Higher the number of vector plasmid, greater the product. That what is our product? Yeah, protein. Okay, so the protein from the gene of interest we will receive after the whole process. Okay, so what is the relationship? Yes, both the copy number of plasmid and recombinant protein are directly proportional. Clear? Then next question. Can exonuclease be used while producing a recombinant DNA molecule? So what is exonuclease? Yes, it's a type of restriction enzyme. So why can't we use exonuclease while producing an rDNA? An exonuclease cannot be used while producing rDNA. This is because it degrades the DNA. It cannot produce DNA fragments with sticky ends. That's why we are using endonuclease. Clear? This is a direct question. Then third one. What are the features of plasmid being used as cloning vector? Which are the three characteristics? It should have an origin of replication. It should have a selectable marker. It should have restriction sites. Then next question. What are competent cells? What does the word competent refers to? Answer. The competent cells are those that allow the foreign DNA to incorporate into the host by slight alteration in the cell walls. So competent means the ability of a cell to intake the foreign DNA. Okay. The next one. What do EcoRN1 refers in the enzyme EcoR1? So that we discussed already, Eco refers to the species from which it is taken. So for example, Eco, Escherichia coli, and R refers to the strain, and 1 is the Roman letter that states that it was the first enzyme isolated from the strain of Escherichia coli. Clear? Then, why are proteases added while isolating DNA? Why? Yeah, proteases added to degrade the proteins so that they do not interfere with the downstream DNA treatment. Okay, so in the first step, while we are isolating DNA, the same step we are doing in our laboratory. So it is a major question for our practical examination. So in that condition, why we are adding protease enzyme? Protease are added to degrade the proteins. That's why the name proteases. Okay, so that they do not interfere with the downstream DNA treatment. Then, seventh question. If the denaturation step is missed during PCR, what would be its effect on the entire process? So actually, what is happening during denaturation? If the denaturation process missed, there will no separation of DNA strands. The primers will not anneal to the template strand, and eventually amplification of DNA will not occur. Then name a recombinant vaccine. Hepatitis B vaccine is a recombinant vaccine. Okay, study this. Then, how is plasmid of Agrobacterium tumefaciens modified to convert it into a cloning vector? The plasmid is a tumor-inducing plasmid. Okay, so the genes responsible for its pathogenic nature are either removed or altered so that it does not harm the plants and only delivers the gene of interest. Okay, so while studying the applications of biotechnology, we will discuss more about this. Okay, Then the 10th question, do biomolecules such as DNA, proteins exhibit biological activity in anhydrous condition? Okay. Answer, no. Biomolecules do not exhibit biological activity in anhydrous condition. Why? That's why life does not sustain without water, isn't it? Then short answer type question, question number 1. What do you understand by gene cloning? A direct question. Gene cloning or DNA cloning is the process in which gene of interest is copied out of the DNA extracted from an organism. Then the process of gene cloning is carried out in the following steps. First step, isolation of DNA fragments, then selection of an appropriate vector, then the isolated DNA fragment is incorporated into vector, then the recombinant vector is transformed into the host cell, then recombinant host is isolated. Okay. Then this diagram is very important. Okay. Name the regions A, B and C. So you study this diagram well. 5 times you draw this diagram and label the parts. Okay. So these are the cloning sites BAM, HI, PST1, ampicillin resistant gene C. Study it thoroughly. Then again from a diagram from textbook is given here and identify the steps A, B, C and diagram. So what is this reaction? Yes, this is polymerization. A is denaturation. The DNA strands are treated with a temperature, higher temperature, 94 degrees Celsius, and strands are separated. Then annealing. The primers anneal to the complementary strand. Then extension or elongation. The DNA polymerase facilitates the extension of strands. Clear? Next question. A gene was being ligated using DNA ligase to the plasmid vector to prepare rDNA during bacterial transformation. Just imagine that step now. An exonuclease were added to the tube accidentally. You know that we have to use endonuclease, but accidentally we add exonuclease. How will it affect the next step of the experiment? There will be no effect on the experiment, isn't it? This is because rDNA is circular and closed with no free ends. We know that exonuclease makes cuts on the ends, isn't it? On the free ends. Therefore, the exonuclease will not degrade the DNA. Simple answer. Then, what would happen if the restriction enzyme do not cut the DNA at specific recognition site? What would happen? The fragment obtained will have no sticky ends and hence the construction of rDNA would be difficult. And the sixth one, how was a DNA viewed on an agarose gel? The DNA is stained with ethidium bromide which intercalates the DNA strands and give orange bands on exposure. While explaining, I showed you one diagram. Do you remember it? Orange bands, exposure to ultraviolet light. Thus, one can view the DNA on an agarose gel. Next question. What would happen if a plasmid without a selectable marker was chosen as a cloning vector? What happened? We can't identify non-transformants, isn't it? A selectable marker helps to distinguish transformed cells from non-transformed ones. Children, do you remember blue and white colonies produced after the process, and the white colonies we can remove easily. I added one diagram while explaining. Do you remember it? If the cloning vector does not have a selectable marker, it would be difficult to select transformants. Then, how are competent cells prepared by the action of calcium chloride? You know that the divalent calcium ions create pores in the walls of bacteria and facilitates the uptake of foreign DNA by bacterial cell. Then the last question of short answer category. A mixture of fragmented DNA was run on an agarose gel. The gel was stained with ethidium bromide again, but no bands were observed. What would be the cause? The DNA must have degraded by nuclease, or the electrodes are placed in the opposite direction of the gel assembly. Consequently, the DNA molecule move out of the gel, isn't it? Then maybe ethidium bromide was not added sufficiently and hence DNA was not visible. Then long answer question category. First question, what is the role of Agrobacterium tumefaciens in plant transformation? We know that it's a plant pathogen that infects crops such as tomato, sunflower, cotton, soybean, etc. It causes Crohn-Gall disease in plants which are induced by TI plasmid or the tumor-inducing plasmid. So the TI plasmid incorporates a DNA segment called tDNA into DNA of the host plant that we discussed. The direct question. This tDNA causes tumors. Then what is bioreactor? Children, the diagram of bioreactor is very important. Okay, draw and study. Explain different types. What is bioreactor? The bioreactor is a large vessel used to carry out. Children, do you remember the diagram I added? The large vessels diagram. The bioreactor is a large vessel used to carry out a biological reaction and to culture aerobic cells for conducting cellular or enzymatic immobilizations. The different types of bioreactors are stripped tank bioreactor, bubble column bioreactor, airlift bioreactor, fluidized bed bioreactor, packed bed bioreactor. Okay. Then what is polymerase chain reaction? PCR again. What are the steps involved? Direct question. Know that PCR is a process used in molecular biology to obtain several copies of a specific segment of DNA. What are the steps? Denaturation, annealing, extension. You should study the steps well. Okay. Last class notes I added its explanations. And what are its application? Yeah, in forensic science, in research and genetics field, and in medicine. Then what are the properties of a good vector? It must be small in size so that it is easily to isolate and purify, and it should have origin of replication. Then it should have a selectable marker. It helps in selecting transformed host cell. Then it should have at least one unique recognition site, cloning site. Okay. Then fifth question, mention any 3 vectorless methods that are used to introduce our DNA into competent host cells. Vectorless method. Okay. First one, transformation. This is the process by which bacteria takes up genetic material directly from the surroundings. For this, the bacterial cells are treated with calcium chloride. The cells are then incubated in ice and then subjected to very high temperature. So this creates pores in bacterial cell wall and foreign DNA is taken up by the bacterial cell. Then micro-injection. Do you remember the picture? While explaining, I added the picture for gene gun method and micro-injection. Here in this method, our DNA is directly injected to nucleus of the animal cell with the help of microneedle. Then biolistics or gene gun method, the cells are bombarded with very high velocity microparticle of gold and tungsten coated DNA. So in this part we discuss both the direct questions and indirect questions. So children, you please study these questions and answer the test question and read the textbook 5 times. Okay. So next class we will discuss biotechnology applications. So before studying that lesson, you should study principles and processes thoroughly. Okay. So we can meet in next class. Thank you.